Hello. How's the time for you at the moment?
It's 5 o'clock here,
Oh,
so
right.
very, very reasonable.
Goodness
Um,
me.
yeah, it's a luxury.
And has so Hung graduated, I think.
Yeah, last or 2 weeks ago. Yeah,
Okay,
done and
great.
dusted.
Okay.
Um,
Is he sticking around for a little
and
bit or
he's got like a short-term contract with like an outreach program for a while.
okay.
But I'm not quite sure what he's planning after that. I don't think he's planning to do research
Okay.
more.
Right. Just easy to stay in touch for things like papers and stuff if needs be. It's kind of useful.
Yeah, I mean, um, yeah, he's certainly still in the building at the moment. So,
Okay.
hi Tim.
Hey, how are you?
Pretty
I'm
good.
okay. How
Nice,
are
good
you?
to hear. Yeah, also good.
How's
Half-term school holidays here at the moment, so
Oh yeah,
supposed to be
lovely.
combining Zoom meetings. We're trying to be a little bit fun.
See, we're just at the point where I start getting jealous of you guys because you're going into summer and
Hmm.
it's.
Yeah, we already had quite a few good weeks now with lots of sun, and it's
Oh,
been very dry, which is not good for the farmers and nature, but it's been
yeah.
nice. And now we're having a bit of rain again, which is good.
Oh, see, Julius has been visiting for the past 2, 3 weeks. And it's rained for like 10 of the 16 days he's been here. Like it's been so annoying.
Hi, Wendy.
Oh,
Hi, we just came back from Brazil, so we're still having a
Oh,
little bit of jet lag.
huh. What was
I
it?
will just stay here, Wendy and Dunya.
That's fine.
I already started it up here. Yeah.
What was
I
the
wasn't
Brazil
actually,
thing again? Sorry, I forgot. I forgot the Brazil thing.
it was
Yeah,
kidnapped, right?
it was the International Society for Human and Animal Mycosis.
Uh-huh.
So I presented there the diagnostic scheme we developed for WHO, and we had a poster on mycetoS.
Oh, that's right.
And there was a lot of other posters on drug discovery for fungi using the MMV boxes, but
Hmm.
not so much, nobody else on open science, I think. So
Okay, great.
And I spoke to Melissa there. And Melissa works on Sporo, and she's planning to stand a student here so that they can screen on Fennery moles to see if they will have activity against Sporotrix too. Sporotrix is another uh, fungal neglected tropical disease causing similar lesions as mycetoma. So, and there's a large outbreak in cats in Brazil.
In cats.
Yeah,
Oh,
so the
And does
cats
then it pass to humans?
and they pass it to humans.
Oh,
So
oh dear.
they went from I think a few hundred cases per year to 10,000s of cases a year now.
Oh, sheesh.
goodness.
Because yeah, it's difficult to eradicate in stray cats because you cannot vaccinate a cat or treat. Well, you
Mm.
can treat cats, but not if they are stray. So
I keep forgetting about the the small box that we're making in our lab of tRNA synthetase inhibitors, which we're having screened. They were designed for malaria, and we're having them screened against everything else we can think of. We must send you some for screening because there's about 20. You know,
yeah,
we've
that's okay.
yeah, yeah. We've made essentially the core compound, you attach an amino acid and you make it selective for that tRNA synthetase. So that you can get the full box of all the different amino acids, and we're trying to assemble a box and send it out. So must do that for MycetOS also.
Yeah. So I think I heard from Dunja there was no agenda.
Yeah, I'm not sure because I remember we were a bit confused, I think, last meeting. So I think you ended up chairing that, even though Erasmus wasn't supposed to,
Yeah,
perhaps. So technically, Erasmus is chairing this one, and you said's taking minutes, but I don't mind.
yeah, I think it has been rotated a lot. But what we can also do is just simply update each other, because I think there's also not that many people around at the moment.
Mm.
And then make a full agenda for next time, so that we officially, because
Get
I
back
think,
on track.
yeah, because I think at this point in time also many manuscripts have been submitted, I guess. So we're rounding up a few series anyway. And maybe it's good to start a discussion again for next week, for next month, on which drug target we should focus on, because we did that 2 or 3 months ago. So people could have thought of it, and that we posted on GitHub also now after this meeting, so that they can really think about it for next for next time.
Yeah, okay, that's good.
Because well, the new season starts with new interns, so we could, even if you didn't secure the funding yet, we could always try to get 1 or 2 proteins made here by an intern, starting from September onwards, when the new when the new round of interns starts at our site. And we made proteins in the past, so the methodology is here. So if we know by September which protein we target, we can design the plasmid and let it be made by an intern, so that at least we have something to start from.
The Seattle Structural Genomics Group for Infectious Disease, who do a lot of the crystallography work, they do make proteins. Their primary thing is to crystallize the proteins so that you can have a look at what they look like. But they make proteins, and I don't know whether we have approached them about proteins they already might have that are from Madurella. I don't know if we've asked that question even. I can't remember
I
if we've
don't
been
think they ever made a protein from Madurella.
You don't think they've made one? Did you say?
Yeah, because why would they make a protein from Madurella?
Yeah, and no, sorry, I think it just there's no one has asked them, and no one has. Maybe it hasn't just been on the list, but technically speaking, it's an infectious disease, and we're open science, and that's the 2 boxes ticked.
Yeah.
So it could be. I mean, I can do that because I think we've asked them about something else already. I can do that and ask them if they have any already, or if they'd be willing to make one.
Yeah.
Yeah.
Yeah, because for me, I think the 2 kinases are still the best bet, because it's new and it is overlap with other fungi.
Yeah.
Yeah, because also in Aspergillus, those 2 proteins are there. I think they're also there in Candida.
Yeah.
And if we look in the future, nobody is going to make a molecule just for mycetoma.
Yeah.
But if we can show that it's also there in Aspergillus and in Candida, and in Cryptococcus maybe, then you have a much broader market.
Yeah. Okay. Action on me. I'll go and approach them and see if they could do that.
Yeah.
I can also just quickly update on the the plan of revisiting the target list would be really good for next time. Yeah. So we have a month to try and think about that and see if anybody relevant wants to come and those things. So yeah, I agree. Let's do that. Just quickly on the manuscript, the Phanorama manuscript. So it was bounced from ACS infectious diseases, and they offered a automatic port over to ACS Omega, which is their general journal for everything that's kind of methodologically sound. And so I've submitted it to Royal Society of Chemistry and Medicinal Chemistry, and waiting to hear back. But nothing has come back yet. So that was a couple of weeks ago, I think. So hopefully that's gone into review and everything. Just on the ACS Omega thing, that's one of those journals where they don't assess importance or significance. They just assess whether it's correct. Like this was the model that was pioneered by PLOS One, which is where everything goes if it's fine. And I guess just in terms of the manuscript on the kinome stuff, which seems to keep getting bounced by journals, that is always an option, that you don't have these journals.
Yeah, I put it now
way.
at PLOS NTD, because I think that's a little bit more focused to NTDs.
Right.
But the kinase manuscript had a problem that, well, Helion didn't accept that we added to co-authors, and then they didn't accept it at all anymore. But Helion is also a little bit broad, so but all the specific journals for tropical diseases or infectious diseases didn't want it. Well, the obvious suspects, and then I went to PLOS Pathogens, and they said it would be more suited for PLOS NTD. And PLOS NTD is still a Q1 journal, so I decided to go for that.
Yeah. Okay, great. Any news on that, or is it gone in, do you think?
I think it's gone in because I didn't get anything that it was not sent out further. But I didn't check yet because I submitted, I think, on Sunday, so last Sunday. So I still needed to check. And I still need to upload the data from MA on the OnCode library. I was planning to deposit that also in the GitHub web page, because we screened almost 6,000 compounds there.
Wow.
And we only got a few hits. But again, those were kinase inhibitors. So I think it's good to display the data there. We did screen it a little bit different than what we normally do for MycetOS, simply because it was too much to screen with the MTS-based assay. So we did it with luciferase and at a lower concentration to narrow down the hits. So only at 2 micromolar instead of 125. So therefore, I will upload it as a separate page in the database. So it will not be the same as the MMV compounds. But all the FDA and EMA approved compounds are in that library. So it's good to have that data there anyway.
Right. And so you said there were a handful of hits in there.
Yeah, there were only a handful of hits.
Okay, if you're able to highlight those so we can have a look at the structures and see what they say.
Yeah, so the majority was azoles. So we left them out for further analysis. But the rest, yeah, they are in the database, and I will make them yellow.
Cool. Okay, great.
Yeah, so I will upload that too. And that one is also submitted at the moment. So it's a JAC, Journal of Antimicrobial Chemotherapy.
Great. The benzoxazoles manuscript, which is sort of shuttling around a few of us. I don't have an update on I need to read I said I'd read the whole paper now because it's nearly ready.
I read through it in the during my flight. So I will upload it after this meeting. So I sent it to my own email, and then I will upload it. Okay. But I think the part which is still missing is the chemistry part a little bit. Yeah. And I also put in a comment. I'm also fine if I'm not the last author because the people from New Zealand also did a lot of it. And what kind of journal they think most suited. Because if there's going to be a lot of chemistry in it as well, then maybe it's not necessarily like a manuscript for Journal of Antimicrobial Chemotherapy or something like that. So maybe then it will be more a chemistry-based journal. But yeah, that's maybe also something for you to look at on the manuscript, what you think, because it's a little bit in between.
Yeah, I still have to just tweak the big scheme in response to the color scheme that you sent through, which is fine. I'll do that. And I just have this. I just really want to try and see if I can merge everything together because yeah, it's a weird diagram with two halves that are very similar, and I just want to see if I can somehow put them together because I think that would be more informative. But I need to find a couple of hours just to do that.
Yeah, and Andrea will send this Thursday 10 extra compounds. I'm not sure where they fit in the scheme, but we can screen them rapidly, especially the in vitro part, and it's larvae season now. So if needed, we could add that to the screening if it's needed for the SAR. For Mars PhD, I decided in the current draft for microbiology, the draft is fine. So that could fit in her thesis so that she doesn't have to wait till we finally send it out for publication. But if you and Andrea think that screening those 10 extra compounds will give essential data on the SAR, then we could also decide, okay, in this current form, the manuscript is okay. Let's add the data from these 10 extra compounds. That will be done during this summer and send it out for publication then. So it depends on how essential those compounds are for your SAR. Because I cannot do that.
Yeah, I'll take a look at the structures and see how they fit in. Yeah, I didn't think that was even a possibility, but if it is, then I'll take a look, and see if it's worth holding out.
Yeah, because at this moment the manuscript is nearly there, but it doesn't mean that if these 10 are very essential, it will not change much on most of the results. It will only be adding a few results in.
Yeah. Okay.
And then Dunya can screen that, and then we simply add her as a co-author somewhere. As well, because Ma is already gone from our lab.
I'll also see if I can find any information about things like solubility and in vitro microsomal clearance and stuff like that you tend to get for the in vitro stuff you tend to get for early stage drug discovery. Because for the phenaramols, it was interesting that we tried to get a few measured in the Dundee thing that we did. But the data for those compounds there was just one compound that actually went through. The others had issues with them that meant they weren't screened. But then you look back at the other papers that have been published on the phenaramols already, and there's a bunch of data there already about whether they're any good, which you can draw upon. And I assume that there'll be something like that for the benzoxazoles because they're pretty common motifs. So I'll see if I can find something about that too, just to sort of indicate that as a class of compounds, they're promising or not, or, that kind of thing.
Yeah.
Okay, sounds good. There was also the series 2, thiazoles paper. Clementine, I guess you were thinking about putting,
I just realized that I forgot to I sent some compounds earlier this month, and I normally send an email, make an issue when I do that, and I appear to have forgotten to. So I'm not sure if they've arrived at Erasmus, especially if you guys have been away for the past week. I feel like that's possibly when they arrived. So
the second round of compounds, you mean?
I sent them apparently on the 1st of May, so I'm not sure when they would have arrived, but
with the card you sent, right?
Yes. Okay. Yeah, you did get
so
that. Okay.
I
cool.
did dilute them already.
Great.
Calculations, put them in the same Excel file you sent first time. And Tessa was supposed to screen them last week while I was away. So I need to check with her if she has any results left right now. The first compounds that you sent in March, we have already done, and we only need to do one in the larva. And
is that the
773, I believe.
Yes, yeah, that sounds right.
So we need to update that in the database. I don't think we did that yet, but those were screened.
Okay. Yeah. Well, if you let me know once you've put them on the master list, then I'll get around to them soon. Because basically with the paper, I was kind of debating whether to wait for that data before trying to share the draft I have or not. But if they've potentially already been screened last week, then I think I will,
last week
put that in.
The first repeat. So I think
that's all right. But
yeah,
I think those compounds, I mean, they're not likely to dramatically change the story. But I may as well just wait a few more weeks and hopefully I'll share it maybe before next meeting. But I mean, I have a rough draft of it at the moment. And the main thing is deciding which compounds we don't do or don't include because, and I can see Peter Rayner has just joined. Very convenient timing. But there are some compounds, for instance, where we've got one or two things that have the same core, and I can't really make any claims about trends or anything when we've just tested one or two of them. But I don't know whether it's worth just putting them in as a kind of just having that in the SI as here's a bunch of other ones we tested, or how to format it, basically.
Yeah, I mean, it's interesting, isn't it? Because for compounds that don't really fit, it feels like you're never going to publish those in any other way. But you may as well just try and put them in at least in the SI and say, well, there's some others, but we're not highlighting those in the paper. That I think that makes it simpler, and then you have the main stuff in the SAR diagrams, and then others. And I think in the phenomenal paper we did that pretty nicely because you had the table of everything.
Yeah.
Well, you had a list of everything we did, but not all of those made it to the manuscript. So I think that's the way to go.
I mean, if you'd like, I can show you the kind of general ideas, the general way I've structured it so far. But basically, we definitely have some that I can't figure out how to put them into a table, but I would like to include them, which is where we've looked at different heterocycles and stuff like that on one side, the one that the 7731 that Junior just mentioned. But yeah, then we have a whole set of semi-random, they're not random, but it's very hard to collect them in a way that makes sense. And also they didn't have any activity. So I think we'll end up just putting them in the SI somewhere.
Yeah, I think that's the way to go. I mean, so if you have a draft of it and you've got, version 1 of the sort of in vitro SAR potency type diagrams, then I mean, yeah, share that so we can have a look. And I mean, don't worry, it's gonna go to version 10, right? So it doesn't matter if they're no good and you're not happy with them, but it just starts giving a sense of what the themes might be of and how we're going to split up the diagrams and what we're going to say.
Yeah. Okay. Well, I will. I mean, I'll tidy it up a little bit more because I haven't looked at it in a month or longer, and then I'll share that. Because the other thing which I still haven't gotten around to, but we talked about it last meeting, was getting the log D calculations and asking Louisa or the DNDi to do that. I wasn't sure because we've obviously, we've made and screened however many, 100 aminothiazoles or something. I wasn't sure whether I need to compile all of those or if I should just kind of, obviously we can't just choose the ones that worked because
Clementine, for them it doesn't make a difference if it's 1 or 100 as long as you provide them with an Excel data sheet.
Yes, so I'll just copy and paste
the program.
Okay, that's good to know.
So I would do all of them.
Yeah, I think that's right. It is interesting. I mean, just as an aside, the calculations of solubility, I mean, they're just woeful, really, really bad correspondence with reality.
Mm-hmm.
So I think we do need 100 and see roughly what the message is. In other projects, we've had solubility experimentally measured for small molecules, I mean, hundreds of them measured for small molecules. So you can actually do the plot, of measured versus calculated. And it's a scatter plot. There's very little correlation between the calculated and the measured. It's really interesting. And the reason, I mean, it's just really hard to measure solubility because you have a really pure sample and it's usually on a small scale and you've got to have the right equipment.
Yeah.
But don't worry about the specifics so much. It's much more about the kind of very rough trend that we see.
Okay, well that's I've kind of been putting that off, but if it's just a question of copying and pasting, I can do that quickly. What else have we got about aminothiazoles? Oh yes, did you ever find the ADME data? I know we only sent a handful of aminothiazoles for that, but
I've got it and I haven't extracted it because I did it for normals and not for series 2. I'll see what's there.
Okay.
Yeah. It won't, it's funny though, it won't be a lot. I'll see what's there. And if it's not a lot, then we can again see if we can dig in the literature and see if we can find something about these compounds in there.
Okay, that sounds good. And I will try and send that draft or share that draft on GitHub soon. Okay. I think that's basically everything we have to say. I mean, we'll probably make a couple more just because it's already kind of in the pipeline with our students. But also, if there's anything else that is more interesting or more crucial to work on, we have a bit of capacity to change what we're doing as well.
Yeah, so therefore I think the next meeting is very important. That's why I also will upload the 6,000 compound data, because I cannot judge which molecules are easy to make for students, but it might be good now that the aminothiazoles, you're preparing a manuscript, to look into that, which series did have some effect on the larvae or which compounds, and which compounds probably have a novel mode of action. So that would be, in my respect, 2 criteria we should look for. And then the third criteria, if you would like to make it a student project, is how easy are they going to be made. So maybe a compound which is very potent, which is very difficult to make will be less priority for this kind of project than the one who does have effect, might be a little bit less potent than the others, but still show some potency and is easy to make.
Yeah, absolutely. Because the ease of synthesis is definitely the biggest barrier that we have with what we're doing here at UCIT, because I am not supposed to be making any more compounds for my thesis.
Yeah. So, but I think that will solve also for the others the question, because we could continue with the aminothiazoles, but if so far we didn't have that much success with the new molecules, but maybe another series which would be equally easy to make has more potential, because I think for the students it would also be nice if we show that the compounds actually work.
Yeah, actually it was very nice. We had a student who's interested in doing, kind of an internship-y thing, basically like a project where they do lab work. And she was interested because we shared the results that you sent us most recently. We shared with them, and therefore she was reminded of having worked on it previously and was like, oh, maybe I want to do that for my project. Because I can't remember which one she'd made, but she'd made one of the ones that was good. Like it had worked really well.
You didn't screen all 6,000 of those in the larvae, did you?
No, we can't. No, we don't have the capacity to do that.
I mean, the actives.
We screened, I think, in the end, 11 in the larvae.
Oh, great. Okay, fine. That's great.
And we had 27 hits, of which I think half of them were azoles, and we already knew that the azoles don't do very well in the larvae, so we excluded them, because also that would not be a new hit. And in the clinical trial, we already saw that the most potent azole is not better than the azole they currently use. So we were more interested in the ones with a novel mode of action, and 2 out of those 11 were toxic, so we couldn't screen, and 2 out of the 9 we screened were active in the larvae, and they both were linked to the NF-kappa beta pathway in humans, but that's not present in fungi. So we try to find what the potential mode of action could be, and we found out that the velvet proteins in fungi have a completely different amino acid structure, but a same folding pocket where the drug binds. So that also makes a very interesting target because it's a regulator. It regulates many cellular processes, and it has a very different amino acid structure. So that would mean that you could make it more fungal specific because the velvet proteins have no orthologs in human, but they regulate all kinds of things. And at least 2 of these molecules most likely were binding towards that target. At least they had antifungal activity, probably also some influence the inflammation system in the Galeria larvae. So we don't know if the prolonged survival was mainly because of that antifungal activity or also because it influenced the immune system. But they did work, so that might also be interesting to look at.
Sorry, what proteins were there?
Sorry?
What proteins for the minutes?
The velvet protein. So velvet like in clothes.
Like the fabric.
Yeah.
Okay, cool.
But that protein might also be an interesting one to think of for designing new molecules because it's in every fungus and it's fungal specific. And it's a regulator influencing many cellular processes.
Very nice.
But yeah, we couldn't prove that it was really the velvet protein because we didn't have a knockout. But yeah.
Sorry, just circling back to the aminothiazole. So the Whitgift school came again to us a couple of weeks ago and had made a compound successfully for that. So there is one more incoming. But they tried to make another compound, which is a bit more difficult. So climatin is one with a nitrogen, and it's got a methyl group on the nitrogen, so it's more difficult to do the coupling because it's an N-methyl. And that hadn't worked very well, and we're trying to sort of figure out why, so they can maybe try again or maybe give us a compound to purify and that kind of thing. But that would be a nice compound to add to the paper, actually, because we don't have any others, I think, with the alcohol and nitrogen.
So how are they? How were you guys making it with the N-methyl?
I think they bought the material with the aromatic ring NH and methyl, and they were trying to do the displacement.
Oh, okay. Because look, ours didn't turn out to have worked as well as I initially thought, but we were doing, like, we took the finished compound and then we were just methylating the N with formaldehyde and formic acid. And that was a student doing it. And what we established was that we think he made the imine and then it didn't reduce all the way. So I was going to try and reduce it myself, but I haven't gotten around to doing that.
Okay.
But that was what our approach was.
Oh, that's interesting. I mean, so that, yeah, it may be that we might be able to fish out the compound from one of the crudes, or we just take the precursors and try it ourselves as another approach. It might be nice to try and get that compound, but we'll see. We'll see what we get when we've done the LCMS of all the samples that they gave us. But I think there's a separate piece of work here, which I'm hoping that Erica at UCL can help push along, which is about the educational bit here and about the how-to of getting students involved in projects like this, because, they've done quite a bit of work on this now and have enjoyed it and have lots of nice things to say about getting involved. And I think we as a team are pretty good at getting student groups involved in projects like this. And so if we can in some way put out a little how-to about how easy it is for people to work with schools to get chemistry done for real projects, it would be quite interesting. I think that's separate from just the MedChem paper, right, where we just got the data. I think there's two things going on.
Yeah, and actually, I also liked giving those lectures to those students because you get questions you never get, and makes you think yourself too.
Yeah, you do.
We, as in Alice and co, are trying to put together, well, we've put together a paper that is about that, but from the perspective of where we're doing it in the university, which is different to what you guys do with Whitgift. But it's stalled at the moment because we did it with a high school, but as an outreach program with them coming to us. And there are some like things to do with ethics with students in publications. So it's kind of hit a bit of a wall, but hopefully we'll get over that wall soon. And we can, share it.
But there's also a big paper that stalled about series 4 in open source malaria, which needed a huge amount of work on over multiple years. That's a beautiful paper. It stalled somewhere, and it'd be great to get that out because I mean, in terms of scalability, that's obviously better even than working with schools.
That one I know, I know that the student who's working on those for Alice, and he's made quite a few of the compounds. I obviously don't know them myself, but I think it was a little unclear whether it was like when I last talked to him about it, he wasn't sure whether it was just compounds that Alice had made that were missing or whether there were other ones missing as well. Because, obviously when you're looking at work someone else has done, it's a little hard to figure out the system. But certainly they've been he's made a bunch of stuff recently. I just don't know how many he needs to make for that to be published.
Just aware Peter's joined us. Hi, Peter. Did you want to say anything about the phenothiazines and the stuff you were working on?
Yeah, so we had our project's just finished up this last few weeks. They made a whole load of aminothiazoles with different linkers, and I think we sent a few across earlier in the year. We've got some more, but whether they are worth doing anything with, I guess we'll wait for the results of those earlier ones.
Those compounds you tested, and unfortunately, none of them were potent at 2 micromolar. I will put the results on the master list today or tomorrow.
Yeah. So we've got a few more, but it's probably not worth doing much with. It could be, but we do have a few more versions of those on the series 2 things. On the phenothiazines, we've got a few, but the synthesis of the ones that we're targeting are really quite challenging for students. So they're not great, to be honest. So trying to make those with the sort of that diethylamine type or ethylene diamine type linking is really hard without just using nitrogen mustards and things like that. So for students using it, it's really quite challenging to make those tails on there. That's where we're at with. We've got some phenothiazines. We've changed the core a little bit, but whether they are worth doing without that link, that tail on, is the other question. So that's where we're at.
Just when you post the data or have a conclusion, if there's some way of highlighting that's been done, either with a mini spreadsheet or a ChemDraw diagram or something, just so that we can look at the structures and any lessons that we should have about the activities, just as a flag, because adding it to the master list is the most important thing, but sometimes it gets lost. So just ping someone that here's the new data for this thing has just been posted and it's there.
Yeah, and we will also color code it again so that people can see that it's new. The thing is, if the chemists look at the data or have looked at the data, could they then uncode the color? Because otherwise everything turns yellow and then the purpose of the color coding will be lost.
Yeah. Okay.
Yeah. So that's where we're at the moment. We possibly have another group of students in September if it's useful to have another group of students, but we're pretty flexible on what we can make. But yeah, the phenothiazines, they're hard, is what I'd say. for students to do and get something successful out at this point.
I mean, yeah, speaking for everyone here, I think it'd be great to have them make a compound for a series that we think is important in September. Obviously, you need some lead-in time, so you know what to order and how to design it and stuff. If we end up coalescing around a target, a molecule class that is difficult, then the other option there is to work to synthesize, gram scale amounts of precursors that we can use for the project, or we provide a precursor and another step is done, or difficult reaction is optimized. But there's always things that can be done, even if the target class is tricky.
Yeah. So we've got quite a bit of data on making some methods to these things, but not the actual compounds themselves. So that's been good. And it is the sort of thing that over summer I might be able to get in the lab myself and make some of the more tricky things, just to see if we can get a couple of hits that prove or disprove something. But yeah, we can certainly do that.
Cool.
Yeah, so maybe that should also be the action point for next meeting, so that we have 2 action points. Actually, the action will be which direction we go for the upcoming time. So one is the protein target, the other one is the drug target, which students can participate in. And then we will make sure that we upload most of the data today, what we have, so that everyone can take a look on what novel hits there are. And shall I, with the color coding, I can also make the ones which were active in the larvae, for instance, pinkish or reddish, or something which really stands out, or orange. So that those are the ones which are the most interesting to look at, because if they are only in vitro active, that's nice, but it might be less interesting than those which are also show in vivo activity.
Yeah, definitely. That would be great.
And it could also be, especially for the student project, we do have the historical data, of course, of the MMV boxes, and there were more compounds active in the larvae than what we focused upon. So if one of these series or these compounds are relatively easily to be made, we can also look back at that. So that we simply have the compound list which all showed some activity. Because we do have also Ibrexafungerp, which is just published, which was active against Madurella and Falciformis spora, but that's a commercial drug, so I don't know how much we can do with that structure and how much is patented.
Could you? I missed that. Would you be able to dump the link or the DOI for that into the GitHub?
Yeah.
I missed that. Yeah, it depends, doesn't it? It depends if it's out of patent or if you can move around the structure and change it enough. Yeah, difficult to know.
Yeah. I really don't know if that was, if that's still on patent. I didn't check.
Okay. Yeah.
So if we're going to have a meeting, well, I mean, we obviously have a meeting scheduled next month, just so that we're all clear, who is meant to be the chair and etc. for that one. So I've just made an issue for it so that we've got one already, but I'm just trying to find the meeting roster where it says who should be what, but
it's issue 2.
Sorry?
It's issue 2.
Issue 2, let's see.
At this stage, it's you, Clementine, and we're minutes.
Okay.
We've just gone the other way around, haven't we? I mean, don't mind. Don't mind how we do that.
I think I'd prefer to take minutes rather than chair for that one.
Right. Why don't we just switch them around then? And I can chair and you take minutes.
Yeah, that sounds good to me. I'll put that in here.
Don't mind. It's a midday one for us, so very easy.
Yeah. And yeah, that means it'll be 9 or 10 for me, so I'd definitely rather not share.
Yeah, okay. Okay. All right, that was everything I had.
Just a very minor thing, but Junior, if you have a poster, do you mind sharing the PDF of it with us and we can put it on the GitHub
Yeah.
somewhere
Yes.
so that we can direct people to it if it's relevant?
I will also put a poster we had on the WHO meeting on I didn't post it there yet. I will post it there as well.
Great.
We have the MycetOS working group, right, Wendy, this week coming up.
Yeah.
The CDC thing.
Yes, I know. It's
On a holiday.
for us it's Ascension Day, but I will try
Oh.
to make it.
Right.
But probably that will only be me then from Erasmus MC. But
Okay.
I spoke to many people already during Isham, so I'm not sure who's going to be there and who's not.
Yeah.
Because apparently it's only for us a holiday, for our region, but the other people had conflicts too. But it's 4 o'clock in our time. So it's probably 3 o'clock your time.
Yeah. I read in the paper the other day about what's happened to Professor Verhaal's lab, and the center has been destroyed, right, from in the war.
Yeah, was it in the news then?
It was in the Guardian Weekly. I was reading about it, and there's a whole picture of it, and saying this lab's been destroyed, and he's having to relocate somewhere to Egypt or something.
Yeah, he's in Egypt, and there was a movie where they stole actually the wiring out from the walls, and that made the lab burn in the end. So there was a burn inside the lab, but the patient files were still there, and for some weird reason, the automated DNA isolation machine was also still there.
Wow.
But many of the other things, and also the -80 was open and destroyed. Many of the other things were destroyed.
Goodness me.
Because he's now planning to rebuild it. But yeah, it's at the moment still not stable in Sudan. So I don't think that will happen in this year. Let's call it like that.
Amazing.
No, because in our news it was also that Port Sudan during the war was relatively stable, but they are hit by drones at the moment as well. So yeah.
Goodness me, very distressing. I wonder if he'll be at the meeting. We'll see.
Yeah, I think so. But from his lab, there are several people at the beginning of the war which were able to join my lab. So Emmanuel finished his PhD here, and he's now in Rwanda. And Rehab and Sahar are still in our lab at the moment. So at least they are safe.
Yeah, safe place to do the work.
Yeah. So a building destroyed is one thing, but it's better that the people are safe. A building you can always rebuild.
Yeah.
But of course, it's a waste of resources, and I rather had no war.
And something which is trying to help the local population, you know.
Yeah. But yeah, I really don't know when this war will stop. There are too many wars at this point in time.
All right. Anybody else want to add any AOBs?
Just a quick one. At the next meeting I won't be around for. We have our external examiners, and in my other job, I've got to look after them for the couple of days. So yeah, I won't be at the next meeting, but be around.
Okay. Cool.
Oh, maybe also a good thing to mention now. I looked at the schedule. After the one in June, it's of course July. I'll be on holiday at that moment in time. For the summer. I don't know if maybe we should also do a poll. I know that probably in the southern hemisphere you don't have summer holidays at that time because it's winter. But who's
right, we
going
have winter
to be around?
holidays.
Yeah,
I'm
who's
away
going
then
to be
too.
around?
For that July meeting, I'm away.
Yeah, me too.
Shall we preemptively cancel it or postpone it?
I reckon we
The
just cancel it.
cancelling's fine.
Yeah.
Okay, easy.
I'll be around for the August one and also for the one in June.
Yeah, June. Yeah, August. I don't know yet. Plans are nebulous. Probably around for that, but I'm not sure. Keep it for the
No, but
moment.
we could also say if July is cancelled and June is difficult, that we relocate in June somewhere, because otherwise there will be two missed. And I think for this point of time, it might be good to have another meeting on the future directions of the target, and especially for the ones who want to let students make molecules in September, to have at least that discussion before.
Yeah, that would definitely be good. Why is the June date difficult again?
June's all right for me.
No, but I don't know if it's essential for Peter to be there.
For the new target.
Because he said there would be exams at the time.
Yeah. I mean, if there's anyone else, if we do want to reschedule it, I think the 1st of July is the next Tuesday, because the last Tuesday is quite early in June.
July. Yeah, that's going. If it's in the morning, it's probably doable.
It would be the same time as this one. Oh no, it'll be the opposite, won't it? Sorry, it'll be the midday one.
But at midday, I have a meeting where I can't go. Yeah, I can't get out of. But it could also be, if it's an ad hoc thing, we could also split it, of course, so that the targets for the protein synthesis will be discussed on the normal meeting, and that we will have a different meeting with those who would like to see which drug target for the students, that we simply add one meeting and only focused on that.
Could do for the July meeting on the 22nd. I'm pretty sure I'm away for that, but I'm pretty sure I'll be back for the 29th, I think.
I have to look in my private agenda because it's my summer holiday.
August, I don't know, but
I think I will be back on the 29th of July.
Okay, that's the day. Or then August 5th is the next one.
Then I'm certainly back.
We could pencil it in for August.
Is Peter, how much do you feel that you need to be at the June meeting in terms of the schedule? Because I assume your students are coming in September. So
yeah, they'll be in September. I mean, in most things, I'm non-essential in life, so I happy to catch up with you afterwards. But yeah, I'm basically those 2 days in June I can't do, and then can do everything from then. But yeah,
I mean, I can do every day as well. So I'm happy to work with what what suits other people. But if you don't feel like it's crucial for you to be there at that point in time, maybe it's simplest to just leave it where it is in June. And if we need an extra meeting to discuss that chemistry, then maybe Matt and, you and me and Matt can sort out a time rather than trying to fit everyone into it.
Let's do that. I think if you have a chat in June, we can see how things are going, and then we can work from there.
Yeah, I'll know better. I'll know dates better by that time in a month. I'll know when I'm around.
Okay, great. Then I'll see everyone in June.
Okay.
Great.
All right.
See you later.
Bye.
Bye. Bye.
See you.
See you.